It's my great pleasure to introduce Dr. Mark Janko, who joins us today from Duke University. You may have heard of that place, up on the coast, sort of. Not quite on the coast, but towards the coast.
Yeah.
Mark is an assistant professor of global health at Duke University. He's a population health geographer and biostatistician, and his work looks at this really fascinating interface between sort of climate and climate change, climate challenges, and the environment and demographic changes. And I'm going to focus on malaria today. Some work in the Congo and Amazon basins, starting off with this beautiful photograph that he took himself. So, Mark, without further ado, welcome and thank you so much. Great to have you here.
Well, thank you so much for inviting me, and thanks for coming and attending online or in person. Especially want to thank the folks who are responsible for organizing this trip. I know that this is hard, and I also know that it's hard to thread a needle between some major holidays, the end of the semester, and so on and so forth. So I really appreciate everyone taking the time to join me today. And I'm going to talk about some of my work in the past and some of the work that I have going on right now in the future. And I'm characterizing it basically as challenges to malaria control in the Congo and Amazon basins. It's kind of a big picture theme of my work. But before I really dive into that, I thought it would be useful to introduce myself a bit more to you all, talk a bit about me, and then turn to kind of give a bit of a background on malaria, malaria transmission, and malaria control, so that we're all kind of on the same page and have a familiar footing from which to kind of launch into these two example projects of mine, these two examples on challenges to malaria control. And the first is work that I did in the Congo on links between agriculture and malaria risk in children under the age of 5. And then the second is my NIH K01. So this is my career award entitled Effectively Linking Molecular Informatics and Network Analytics to Reduce Malaria. It takes place in the Peruvian Amazon, so it has this nice acronym, ELIMNAR Malaria. And it was also useful because NIAID was only funding K01s in the area of data science, and so I thought if I put the words informatics and analytics in the title, it would convince reviewers and the NIH that I was doing data science. And it worked, because I'm in year 1 of that grant right now. So a bit about me. So I'm a population health geographer and a biostatistician, as I said, or as Jim said. And as a geographer, we're a pretty rich discipline and a very diverse discipline. Folks work on, hydrometeorology and things like what happens with major storm flow events. And then there are people who study kind of ethnography and indigenous peoples. And then there are people like me who study health. But broadly speaking, most of us return to some big picture question about how do humans interact with the environment. So a lot of my questions are at the intersection of how humans interact with the environment and how those interactions affect malaria risk. So what does climate and environmental variability, how does that influence our malaria risk, or features that in turn can contribute to malaria risk? And health geography is a rich discipline that goes back many, many years, hundreds of years in fact. And so over the course of time, we've developed a number of theoretical frameworks to think about health outcomes. And one of the most kind of foundational and formative figures in my discipline is a woman named Melinda Mead, who in 1977 kind of took a lot of ideas from her predecessors and kind of put them together and they coalesced in this thing called the triangle of human ecology. And in doing so, she was, as I said, she was building on some of her predecessors' work, in particular Jacques May in 1950, who wrote that today we recognize that disease is a multiple phenomenon which occurs only if various factors coincide in time and space. Now, to all of us, this probably seems pretty obvious, right? Whether or not you study infectious disease or chronic disease or whatever, that we know that there are these multifaceted forces that influence health outcomes. But at the time, this was kind of relatively new, at least in the health geography space. But it also kind of highlights that these factors, need to coalesce in time and space. Factors such as population, our demographics. So much of our health is a function of our demographics, age and sex, etc. Also our culture, our behaviors. So behaviors being kind of the observable aspect of culture. Do we engage in behaviors that increase our chance of becoming ill? Do we smoke? Do we get enough sleep? Do we get enough exercise? Do we sleep under a bed net if we're in a malaria transmission zone, etc. And then the environment, so the habitats that we live in, or the environments that we occupy or are exposed to. That can be the built environment, such as a city and the services that it provides. It can be the social environment, or it can be the natural environment. On top of that, so if we have these kind of three pillars of interacting forces in different ways, one of 25 years after Jacques May was writing, this guy, John Hunter, who was Melinda Mead's PhD advisor, kind of issued this challenge to health geography. And it was kind of pointing at the GIS map-making wing of geography. It was like, hey, we're really good at making maps to describe patterns. Like, things are really high over here and they're really low over there, but really bad at thinking about the processes that drive these phenomena. And so Melinda Mead, who was John Hunter's PhD student, kind of came along and said, all right, well, let's kind of put together this framework that allows us to think about process, space, and time, and kind of a holistic way of thinking about health outcomes as the byproduct of our population characteristics, our cultural characteristics, and our environment. And that's really nice. That's a big picture framework that says, hey, we know how to think about things, but at some point we probably want to test some hypotheses, and we probably want to build some evidence, and we probably want to try and move the needle a little bit in our respective fields of global health for malaria control. And so I'm also a biostatistician. And so the reason why I'm a biostatistician is because it turns out, in my mind, that hierarchical Bayesian spatial and spatiotemporal models are really a good way to try and understand spatial and spatiotemporal processes. They're really flexible, and so this is where kind of methodologically I trained in. That's not to say that statistics is the best thing ever, or geography is the best thing ever. I work in highly multidisciplinary settings. And there are lots of ways to develop evidence and present findings and try and move the needle, but this is just kind of how I'm trained, kind of how I'm built to think and answer problems, answer questions. And most of those questions have to do with malaria. And so just as a bit of background, malaria is a vector-borne disease that fundamentally requires humans to interact with mosquitoes in a common environment. And kind of the caricature of transmission, the most basic story of transmission is that a mosquito will bite you while you're asleep at night, and maybe you have malaria, and they'll extract your malaria parasite. And later on, after they've rested and they get hungry again, they'll bite another person and give them malaria. So really, it really requires mosquitoes and humans to interact in a common environment. So it's useful from the perspective of a geographer interested in understanding human-environment interactions, and I'm making the case here that malaria is well suited to geographic study. Now, there are several species of malaria that can be transmitted to humans, and there are really two of major global health concern, Plasmodium falciparum and Plasmodium vivax. And they're transmitted by female Anopheles mosquitoes. And there are hundreds of species of Anophelines, and dozens of them that can transmit malaria to humans. And so one of the things that I like to think about is when we're thinking about population health and populations and malaria, we actually have two populations that ideally we would like to think about at the same time. The mosquito population that transmits malaria, and the human population that gets it. On top of that, malaria is also a big problem, so I don't work on it just because it's a nice system to think about. But over 3 billion people, mostly living across the tropics, are at risk of malaria in any given year, at risk of becoming infected. And each year, roughly 250 million people become infected, and over 600,000 people die. So it's a big problem. It's a major source of mortality around the world, and we're really trying to reduce that mortality and reduce transmission. And we have a number of tools that allow us to do so. So starting around 2000, there was just this massive scale-up of a set of core interventions to reduce transmission. So the first is long-lasting insecticide-treated bed nets, which act to provide a physical barrier to prevent a mosquito from landing on you if you're asleep at night, and a mosquito is looking for a blood meal in the middle of the night inside your house. But they also have an insecticidal effect, where the mosquito can repel, or the bed net can repel the mosquito or kill it if it makes contact. But there are concerns, ongoing concerns about the effectiveness of long-lasting insecticide-treated bed nets. Mosquitoes develop insecticide resistance, so there's kind of this constant effort to revitalize the efficacy of bed nets with new insecticides, synergistic compounds that enhance the efficacy of these bed nets. Similarly, after a mosquito bites you, it needs to rest after its blood meal. And typically, if a mosquito is biting indoors late at night, it will rest on the inside of your house, the inside surfaces of your house. And so one of the other main interventions has been to spray these indoor residual insecticides, indoor residual spraying, to coat the inside surfaces of your home with an insecticide such that after a blood meal, if a mosquito lands on that surface, it'll die. But here too, we have concerns with insecticide resistance. Mosquitoes also adapt their behavior to avoid dying due to insecticides. They might fly outside the house instead of land in it. And then if you do get malaria, if you're unfortunate enough to get malaria, we also have rapid diagnostic tests. So we really want to understand whether or not you're infected as quickly as possible, such that we can administer therapies. So for Plasmodium falciparum, kind of the mainstay intervention, the mainstay treatment, are these artemisinin combination therapies, which kind of act in two ways. One, artemisinin rapidly clearing parasites out of your bloodstream, and then some sort of partner drug that sticks around for a while, kind of wiping out any residual parasites that are in your system. But here too there are concerns. There are ongoing concerns with the effectiveness of our interventions. One, diagnostic resistance. So where I work in Peru, the malaria parasite has essentially evolved where it no longer expresses the protein that's detected by these rapid diagnostic tests. These are known as HRP2 deletions. This is for Plasmodium falciparum. And then also drug resistance has emerged to every single antimalarial drug that has ever been produced, and that antimalarial resistance always crops up in Southeast Asia, in Thailand, and Cambodia, and Myanmar. And so these things are kind of the mainstays of malaria control, but there are constant challenges to making sure that we maintain their efficacy through the development of new drugs, the development of new tests, the development of new insecticides, and so on and so forth. And over the course of, since 2000, over the course of the first 10 or 15 years since 2000, we made a lot of progress. So what I'm showing here is the deaths due to malaria each year from the year 2000, and we've reduced mortality by about 25%. From over 800,000 deaths a year to roughly 600,000 deaths a year. But since that time, the number hasn't budged much. And so these data are based on the most recent World Malaria Report that was published. I think this is the 2023 report. And as we can see that basically for the last 10 or 12 years, we haven't made as much progress as we need to. Things have stabilized. And in part, this is due to the concerns that I mentioned earlier. Ongoing concerns about durability of interventions, insecticide resistance, drug resistance, diagnostic resistance, mosquitoes' behavioral adaptations to interventions, and so on and so forth. But I want to make the case that we also know that we lack a sufficient understanding of malaria ecology to eliminate the disease. So if fundamentally malaria's transmission is governed by these complex interactions between humans and mosquitoes in a shared environment, we really need to understand these interactions in order to eliminate disease, to reduce transmission, etc. And the way that that manifests itself to my mind is to say we don't fully understand how human environment interactions influence malaria risk. And so this brings me to kind of my first example of some of my past work in the Congo, entitled The Links Between Agriculture and Malaria Risk in Children Less than 5 years of Age. So the motivation for this work, is that agriculture is a fundamental way that humans interact with the environment, particularly in the DRC. So over half of the population lives in rural settings where people are predominantly engaged in subsistence agriculture. So predominantly farmers living in rural areas. But it turns out that this activity may be driving malaria transmission, since small-scale farming produces transient sunlit pools of water, which are the favored breeding habitat of Anopheles gambiae mosquitoes, which are the most pernicious malaria vectors in the world. And so I have an example on the left here of a farmer in the Congo who's watering his crop, and you can see this transient sunlit pool of water just kind of by his feet in that narrow trench. This is the ideal breeding habitat of Anopheles gambiae mosquitoes. We can also get the same effect after a rainfall event, and so on and so forth. So there's a fundamental dependence on the environment. At the same time, the Democratic Republic of Congo has this dual burden of disease. So a third of the population is infected with malaria at any given time, and at the same time, a quarter of the population is experiencing acute hunger. And so expanding agricultural production to increase or to address food insecurity may contribute to malaria risk. So we have this dual problem, food insecurity and malaria. And the solution to one, more food, at the most basic level, I'm not a food security expert, so apologies in advance, may be exacerbating another problem, malaria. And I'm not the only one who's thought about this. So a lot of people have, from a variety of different settings and a variety of different ways, have looked at the relationship between agriculture and malaria. And the studies largely have found that agriculture and agricultural development has been associated with a number of quote-unquote malaria transmission parameters. In other words, they're doing entomological studies and they're saying, oh look, when we find in these agricultural plots, we find more mosquitoes. We may find more mosquitoes resting indoors. And therefore we might think, well, there must be more malaria. But that's not necessarily the case at all. And for about 20 years, there was this paradox, this thing called the Paddy's paradox, where people were researching irrigated rice fields and finding that there were a ton more mosquitoes and there was less malaria, or not more malaria. And it was this paradox because it was completely counterintuitive. And one of the reasons why it was kind of counterintuitive was also because these studies often lack data on other important factors governing transmission. So thinking back to kind of this triangle of human ecology and thinking about kind of the multiple phenomena of disease. So a lot of entomological studies aren't looking at housing characteristics of the local population. They're not asking people if they're sleeping under bed nets. They're not actually necessarily measuring transmission itself. And that's not necessarily wrong. Entomology is expensive and try and answer the questions that you can. But it does lead to kind of weaknesses in the conclusions that we can draw. On top of that, a lot of these studies are done in a small number of sites, limiting the representation or the representativeness of those findings to a larger country or a larger region, where kind of a level where decisions are being made with respect to, for example, malaria control. But on top of that, the findings may be perfectly correct at a given site. But mosquitoes have different behaviors depending on where they are. They respond to interventions differently. So we know that after 10 years of intense malaria control where everybody's getting a new bed net every 2 or 3 years, mosquitoes start biting outside because they don't like coming into contact with insecticides. And so if this is unobserved, we might have conflicting findings, etc. And so I've stepped into this space with the objective of assessing the relationship between agriculture, mosquitoes, and malaria in the DRC, and in particular doing so using population-based survey data. So many of you may be familiar with the Demographic and Health Survey program, but these are these $10 million surveys that are paid for by USAID, and in this particular survey from 2013 to 2014, they sampled 4,600 kids less than 5 years of age who are living in 331 communities all across the Congo. So not a handful of sites, but covering the entire country. And those are kind of the yellow dots on this map. And this survey was conducted from August 2013 to February 2014. And then by complete coincidence, entomological surveillance was being done at the exact same time in a subset, or in a smaller set of communities, in 6 or 7 communities across several different ecological zones across the Democratic Republic of Congo. And this surveillance number led to 6,700 Anopheles mosquitoes captured across different ecological zones between August 2013 and July 2014. And the sampling was done using something called human landing catch. And it's exactly what it sounds like. You go up to someone's house, the 2 of you, and one of you says, hey, can I sit inside your house for an hour with my shoes and socks off and a straw and a flashlight? And every 30 seconds or so, I'll turn the light on and I'll see if mosquitoes land on me, and I'll suck it up in the straw and put it in a cage. I've done this. I've done this year. And then someone else does it outside, and every 2 hours or so you switch spots to prevent mosquitoes from having some sort of attraction bias. But this is one thing that allows us to understand mosquito behavior, knowing where they're biting, identifying the species composition, etc. And so in this study, I actually ended up having 2 outcomes. One from the Demographic and Health Survey data, the malaria status of each child. So we diagnosed malaria using PCR in the lab where I did my PhD. And then our entomological outcome is whether or not mosquito was caught biting indoors, which we're defining as an indoor biting mosquito. And that kind of harkens back to some of the earlier literature that was finding increased indoor resting densities and increased abundance of mosquitoes in agricultural settings. And then the exposure of interest here is the percent of agricultural land cover within 10 kilometers of each survey cluster and entomological surveillance site. And we picked 10 kilometers for one reason primarily. It's the maximum flight distance of a blood-fed Anopheles mosquito. If there are any entomologists in the room, I know you don't believe that, I don't either, but there's a paper published that says it's the absolute maximum flight distance, and the reviewers decided to believe me. But it also kind of corresponds to, what could realistically be kind of your daily activity space in the rural Congo, where there's not a lot of transportation infrastructure, and your farm plots are probably not more than 10 kilometers or so away from your given house. And then the hypothesis that I have, is that increased exposure to agriculture is going to increase risk of malaria via increased indoor biting. But also that this agriculture-malaria relationship probably varies spatially for a variety of reasons. First and foremost, I measure agricultural land cover. I don't measure the type of crop. So if you're growing plantains and your crop is quite mature and it's casting shade over these breeding habitats, you might not have these Anopheles gambiae mosquitoes. You might have a different species of mosquito, like Anopheles fenestus or something, which would potentially impact your risk of malaria. Similarly, in these 331 surveillance survey clusters, we don't observe the mosquito population. We just observe it in 6. I would love to observe it in 331 sites, but that's really expensive. Doing human landing catch overnight in 331 communities. Okay. And then since we're working with population-based survey data, we have the advantage of being able to incorporate a lot of the other behavioral and demographic and environmental factors that a lot of previous work hasn't been able to address, such as age, sex, bed net use. So Patty's paradox kind of plays out as follows. Well, what happens if the reason why we're not seeing a lot more malaria is because people with larger agricultural fields have better socioeconomic position, they have improved housing, they're more likely to sleep under a bed net, they have better access to health care, and thus lower malaria risk. So bed net use, household construction materials. Additionally, the mosquito population and the parasite population is heavily dependent on temperature and precipitation. And so we're incorporating those data that we derive from satellite imagery as well. And then from the entomological surveillance data, we have temperature, precipitation, the month of surveillance to try and understand seasonality a bit of mosquito abundance. And then we also identify the species. And so this is the statistical mathy slide. And so to test this hypothesis, we're using multi-level probic regression models. And in particular, we're allowing the effects of agriculture on malaria risk in these kids to vary across space. And we do that using something called the Gaussian process, where basically we say everywhere on the map there is a regression coefficient capturing the effect of agriculture on malaria. And if you've ever fit a multi-level model in your life, this is a mere extension of that. Everything that plays out in the multi-level model versus kind of the spatial model is just right here. And so this is one of the reasons why these are pretty flexible, is because there's an intuition about multi-level models that we may have, either through our own work or through collaborations, but extending it to the spatial or the spatiotemporal case is merely just kind of a mathematical nuance that doesn't actually fundamentally change the underlying principles, but it does allow us to make inferences about spatial processes, which is one of the goals that we have in health geography. And so we're allowing the effect of agriculture to vary on malaria risk for the reasons that I mentioned. We don't observe the crop type, we don't observe the mosquito, and so the effect as a result might vary. And so as we move in to talk about the results from this first example of my work, one thing I want to highlight is that malaria exhibits considerable heterogeneity. We have both large areas in these deep orange colors of high risk, but we also have large pockets in these yellow areas of relatively low risk in this case being prevalence. But we also have focal pockets of really high risk adjacent to low risk areas. And so this is kind of part of the other motivation for thinking about effects varying spatially, is because we just observe spatial variation in transmission intensity, and it might not be fully captured by assuming that an effect on an outcome is constant across space. And so the principal finding from this work is that in children under 5, a 15% increase in agricultural cover was associated with risk differences ranging from 0%, so not really any impact at all, to 3%. And these are absolute scales, so it's not a relative risk, it's an absolute risk difference. So if you have a third of the population infected with malaria and agricultural exposure might increase their risk by 3%, I think that's meaningful. On a relative scale, it would be like a 10% increase. And that variability depends on a number of other factors, such as improved housing and sleeping under a bed net treated with alpha-cypermethrin or deltamethrin, which are more effective in the Congo. Permethrin was another major insecticide that's no longer used in the Congo, if memory serves, because it was found not to be effective. And from the analysis of the entomological data, we found that the effect of agriculture on indoor biting behavior increases. So the likelihood or the probability of finding these Anopheles gambiae mosquitoes biting indoors increased as a function of agriculture, anywhere between 11 and 20%, again varying by season and other factors. But I mentioned I was also interested in understanding how this effect might vary across space. And if you look at the panel on the left, this is a plot of all the regression coefficients at kind of every point on the map. And so we see swaths in red here, where for whatever reason, either the mosquito population is different, or the crop grown is favorable to increased transmission, it's associated with higher risk of malaria in these kids than the overall average effect. Conversely, there are these areas in blue where there's much lower risk of malaria as a function of agriculture. And we don't necessarily know why. So one of the reasons why I kind of work in this space is because we can look at these maps and say, well, what might be happening there? And go on and do additional work to uncover the and try and understand what the human-environment interactions are in those spaces so that we can better understand the ecology of malaria and reduce transmission. So, in this first example, kind of one of the main conclusions that I have is that I hope I've convinced you that agriculture is a critical component of malaria ecology and can increase malaria risk in children depending on various factors, and that this effect also exhibits some spatial variability. And since I've published this, there's been some new work that has come out that has kind of resolved the Patty's paradox. So there's this really nice meta-analysis that came out and said, in fact, agriculture does increase malaria risk. And one of the reasons why this is important is because a lot of development agencies have been going to the Congo and saying, no, rice production doesn't affect your malaria risk. Go ahead and use and farm rice as much as you want. But that conclusion is no longer valid based on this research. Now, that being said, you can publish the paper. Unfortunately, this paper is published in the Lancet Planetary Health, and I welcome you all to read it. The results are presented to the DRC Ministry of Health and the Ministry of Environment and Sustainable Development, but these are things that are kind of more or less under my control, right? I don't necessarily get to choose where I publish, but if I try hard enough, occasionally I get lucky, and then I obviously am able to decide where I share my results via partnerships in the Ministry of Health and in the Democratic Republic of Congo. But above and beyond that, one of the things that's been really gratifying to me with this work is that the WHO has a strategic advisory group on malaria eradication. And one of the working papers that they came out with was megatrends that will affect future scenarios of malaria. And my work is cited as part of that. So there's kind of this increasing recognition that kind of land use, land cover, ecology, human-environment interactions are required to eliminate malaria. And my work is trying to contribute to that. It's being recognized somewhat. Okay, let's switch to the Amazon. This kind of just move continents and talk about effectively linking molecular informatics and network analytics to reduce malaria, which is a title that probably sounds nothing like anything that I do, based on what you just heard. And the overall objective of this work is to understand how human mobility contributes to the diffusion of malaria in the Peruvian Amazon. And you might be saying, okay, well, this guy studies agriculture in the Congo, and now he's talking about studying human mobility in the Amazon. What's this guy about? This doesn't make any sense. But I want to make the case that another fundamental way that humans interact with the environment is they move through it. And that human movement is fundamentally a sociotemporal process. So it is uniquely suited to the kinds of questions and tools that I use to answer them. But it's also kind of consistent with theory. So human movement is a behavior, right? Some of us chose to take the bus today, some of us chose to drive, some of us chose to work remotely, and so on and so forth. Human movement is also structured by age and sex. It is highly unlikely that your 2-year-old filled up your gas tank this past week. And then in the Amazon,
Good point.
and in the Amazon, human movement fundamentally depends on the river levels, on the levels of the river. So where I work, there really aren't any roads. So if you want to move around, you're either hiking through the jungle or you're spending some time on a boat. And climate and environmental variability deeply shapes movement in the Amazon, and deeply shapes it right now. In the Amazon, we're experiencing a historic drought that is really affecting human movement. It's affecting our field work. Our field teams, including myself, we would normally take a boat. We spend a lot of time walking along parts of the Amazon River and its tributaries. And on top of that, human movement is a major risk factor for malaria. So as we all know, human movement is a risk factor for infectious diseases generally, and malaria is no different. It's also a risk factor in the Amazon specifically, and it undermines malaria control in a couple of different important ways. First, it continuously leads to reintroduction of parasites into areas, undermining malaria control. So you might really be trying to reduce transmission in community A, but if people keep coming into community A from community B, and community B has a lot of malaria, they will just continuously reintroduce parasites. And if those parasites happen to harbor drug resistance or diagnostic resistance, you might find yourself not staying in a stable state, but fighting a losing battle, where you have drug-resistant parasites come in and suddenly your treatments no longer work. And so if we want to address this problem, we fundamentally need to know, why do people move? Right? So for example, just to give one example of why people move in the Amazon. In the rural Amazon, people lack access to the formal financial sector. So a corollary to this is I flew down from Raleigh-Durham today, and I didn't know any of you before this meeting, but I was reasonably sure that my access to the formal financial sector would allow me to get an Uber, or buy dinner, etc. But when you lack access to the formal financial sector, you need to mitigate that risk. And one of the ways that peoples in the Amazon do this is they'll travel between communities where they have socioeconomic, occupational, or relational ties. And if, for example, they're moving for work and are moving to a setting where they have occupational ties, they might work in a variety of different sectors, like fishing, or logging, or mining, or agriculture, which can put them at risk of malaria. Or they might be taking their kids to school, or they might be going to play soccer. This shows up in our survey data. People like to play soccer in the Amazon. But the consequence of this movement, of these travels, is that these travel behaviors and work settings can place them at risk of malaria. And when they're infected, they can bring infections home with them, causing local outbreaks, undermining malaria control. And so to reduce transmission, it's necessary to understand when, where, and why people move. An intervention to control malaria is going to look very different if you're trying to help prevent, a grandchild who grandma is taking to a school versus their father who's going to work in a mining concession. The interventions will kind of necessarily need to look different, if only in the way that we engage with the community. Since mobility is kind of really strongly governed by kind of social, community, occupational, the network ties that connect people together, the ties that bind us, we need to understand those. Right? In order to have an effective intervention, to engage with communities, to understand how their behaviors, how their travel patterns, how their lives lead them to be at increased or decreased risk of malaria. And then finally, we need to understand how human mobility leads to parasite mobility. I'm pretty sure I don't have malaria right now. I flew down, and lots of people move around, but they don't have malaria, but some people do. And so we need to be able to distinguish those two things, human movement versus parasite movement. And lo and behold, I'm not the only one who's thought of this. A lot of work has looked at not just human movement and infectious disease generally, but human movement and malaria specifically. And so there have been a lot of papers that are really, really cool and really, really good that have used things like cell phone data, or wearable GPS devices, or genomic data, or travel history survey data. Well, cell phone data is great, except there's no cell phone coverage in the rural Amazon. I've tried. It doesn't exist. It also doesn't tell us why people move. It doesn't even necessarily tell you who moves. A household has one cell phone. You don't necessarily know the female or the male head of household who's going from one community to the next. Wearable GPS data, it's really cool, but it's also very expensive. If we're trying to understand human mobility along regional scales such that we can kind of design interventions that kind of capture the spatial scale in which movement is contributing to transmission, we need to be able to deploy tools of data collection that can actually be scaled to those larger spatial and temporal scales of movement. And it also doesn't inform why people move, unless you accompany it with some survey data. Genomic data. Genomic data is really cool. I've been working with some genomics folks, malaria genomics folks for well over a decade. And they do really, really cool work where they go to 2 communities, for instance, and they say, okay, let's get everybody's malaria parasites, deep sequence them, and compare them. And if the parasites sequences are really similar to one another, we infer that movement exists between these 2 places. But it doesn't inform why people move, right? Or who is moving, just that these places are connected in some way. So it lacks a certain context about what's going on the ground. And then finally, same problem, travel history survey data. Oftentimes part of routine surveillance, including in Peru, Brazil, Ecuador, and a lot of places where I work, but it doesn't provide reasons of travel or information about the communities where you're coming from or going to. And so at the end of the day, I'm making the argument that better measures of human mobility are needed. And in particular, we need measures that are scalable, so that we can understand mobility across large regions, so that we can understand how it contributes to transmission. And then we also need better information about the community network ties that drive the timing and the magnitude of mobility, so that we can understand how to reduce transmission with interventions that are specifically tailored to the types of risks that these people who are moving in the Amazon face. And so I'm doing this work in the Loreto region of Peru. So just to orient ourselves, we're in the far northwest of the Amazon region, and the far northwest of the Amazon region has much of the northern Peruvian Amazon making it up. So much of the northern Peruvian Amazon is in this kind of northwest Amazon region, and it's home to roughly 1 million people. Half of whom live in this capital city of Iquitos, which is the size of Miami. There are about 500,000 people living there, and there's not a single road going to it. You can only get there by boat along the Amazon River or some of its tributaries, or you can fly there. Outside of that, it is very rural. If you want to travel, as I said earlier, you are on the tributaries of the Amazon River system. And so much of the population is distributed in these rural communities in the many tributaries of the Amazon River, and it's also where 90% of the malaria transmission in Peru happens. And transmission is largely driven by Plasmodium vivax, representing about 3/4 of the cases, and the remaining are Plasmodium falciparum. And so just to further orient you to this study site, so I'm working along 3 of the major river systems in Loreto. The Pastaza River, which connects Ecuador down to the Marañón River, the Marañón River, which connects to the Amazon River, and the Amazon River, which connects down to Brazil. And I have these in different colors because my K01 is piggybacking off of an R01, and we've already collected a lot of network, or a lot of mobility data along the Pastaza and Amazon rivers, and I'm going to be collecting data along the Marañón River, mobility data on the Marañón River, and additional data on all 3 of these. And so methodologically, if we're trying to understand the community network ties, the ties that bind us, that drive mobility, it might be useful to use network-based sampling to understand network mobility. And the overarching hypothesis that I have for my grant is that communities in the Amazon, through human movement and the drivers of human movement, can be thought of as being organized into networks of communities. So communities don't exist by themselves, but they make part of larger networks of communities. So the community where you go to play soccer, the community where you go to attend religious services, the community where you go to receive health care, the community where you go to buy and sell goods, so on and so forth. And these communities probably have different malaria transmission intensities, different degrees of connectivity to other networks of communities. And so how do we discover this network structure? Well, as I said, network-based sampling to understand network connectivity. And so what we're doing is we're using a respondent-driven sampling approach as follows. First, we're going to select 10 initial nodes along these river systems, these node communities. And we might select these nodes based on their transmission intensity, so high transmission, low transmission, medium transmission. We might select some of them based on whether or not they're in indigenous territories, such as the Achuar indigenous territory in kind of the western part of Peru along the Pastaza River, versus non-indigenous. Distance to the border, right? How far are they away from other border areas, and so on and so forth. And then we're going to go to those 10 communities. And we're going to enroll 4 or 6 key informants, so people who know the community really well, its history really well, its peoples really well. And we're going to just simply ask them over the course of about 20 minutes, where do people go? And they'll say, oh, well, people go to community A. And we'll say, okay, why do they go there? They'll say, well, they buy and sell goods, they attend religious services, they play soccer, they look for work. How many people go for those reasons? each reason. And when do they go? And then we say, where else do people go? And they say, oh, we go to community B, so on and so forth. And then we ask, where do people come from? So a really quick 20-minute interview asking about where people in the community go to, why, when, and where people come from. And then we rank these communities. We do some math on pen and paper in the middle of the jungle, and we follow up. We say, okay, well, this community is the most connected overall for kind of all of the reasons over here, so we need to go there. This community is the most ranked for, buying and selling goods, and this community is the most ranked for, is the highest ranked for some other reason. And so then we get on a boat and we go to that community, and we do it again. And we keep doing it until we run out of money. Which turns out to be about 3 waves of data collection. But what we found is that we actually kind of saturate the network, where once we're in this 3rd wave of communities and we're asking where people go to and from, they keep naming places where we've already been or already collected data. So it turns out that this is actually quite scalable, a scalable approach to rapidly assess how communities are connected across large regions in the Amazon basin. And it turns out that you can do respondent-driven sampling by boat in the rural Amazon, the cross-country borders for the R01, or internally in Peru for my K01. And that's going to allow us to understand human mobility, the drivers of it, the timing of it, the magnitude of it. But it doesn't necessarily tell us whether or not this mobility is leading to the spread of parasites. We can link these data to the routine surveillance system, and we're working on that right now. But one of the things that my K01 is doing is saying, well, we also need scalable tools to understand the malaria genomics so that we can link parasites between communities. And if we're trying to understand human movement across very large distances, we need less genetic information. But at shorter and shorter distances, say kind of like regional differences, we need more and more genetic information because we want to distinguish parasites that are driven by movement versus that are just kind of similar. And so one of the things that my grant is doing is all along the Pastaza, the Marañón, and the Amazon River, these 3 river systems, is working with local health posts to collect dry blood spots from malaria-positive patients. And then using these things called molecular inversion probes to genotype malaria infections. And these were initially developed to overcome the high costs of deep sequencing, but also give similar kind of resolution of the malaria parasite population. So 1,700, 1,800, 2,000 SNPs across the entire genome. So scalable network data collection, scalable genomic data collection, and then to test this overall big picture hypothesis. Is malaria genomic connectivity a function of network connectivity? Network connectivity defined by human movement for a variety of reasons. And so some of the big picture questions that I have in this grant are, how are communities organized into networks based on their connectivity, kind of recalling what I was talking about earlier. And then do community networks, if we can construct these networks, do those networks, when we create them from the mobility data, do they look similar to the networks that we see from genomic data? Because we can basically use similar tools to identify these networks either way. To what extent are they overlapping? What type of mobility drives importation between communities? Is it going to soccer matches or religious services? In some of our sites, there's a big, you see in the surveillance data, big spikes in transmission, 2 weeks or a month after a major religious ceremony. And unless you know that there's a major religious ceremony in that community, you have no idea what's driving this. But this is the type of context that we're trying to understand. And so to answer these questions, I'll be using kind of my hierarchical Bayesian methods, linking kind of this network and genomic data. And it turns out a lot of network methods are kind of corollaries to spatial methods. So when I was writing my K01, you have to learn new skills when you write the grant. So I wrote, I'm going to learn network analysis. And it's hard to write a grant about something that you don't know how to do yet. And so I was like, okay, well, I'm a statistician, and frankly, this is how I would do it. And I started getting really excited, thinking I was going to have this really high-impact paper in a statistics journal about basically just copying and pasting statistics work and saying just use the word network instead, and all the math is the same. But it turns out that these things are called network autocorrelation models, and the math is pretty much the same. So a lot of the tools that I have are translatable to the network data context. But there's also a number of other tools from population genetics and network analysis that I'm working to develop skills and to answer these sorts of questions. And so progress so far, this grant was just funded in June, so most of my milestones are administrative. I mentioned that this grant is piggybacking on an R01 that's spanning the northern Amazon across Brazil and Peru borders and the Ecuador-Peru borders. And the data collection for the R01 is complete, which is great because I'm using it. And we managed to visit approximately 300 communities using respondent-driven sampling approach. So we showed up in 300 communities by boat all along here. And I'll be filling in the gap. I'll start filling in the gap with this network survey in the summer of 2025, up and coming, and then I'm going to be starting to collect the genomic data along the Pastaza, Marañón, and Amazon in 2026, such that in end of 2026 and into 2027, I'll start to have preliminary results about how communities are connected into broader networks and how malaria is diffusing between those networks and within them. And this study is, I'm taking kind of a long view, right? You know, we're trying to understand human-environment interactions and their impact on malaria transmission. We're not doing it simply for an academic exercise. We want to understand how we can improve malaria control because, as we saw earlier, progress is stalled. We need more tools. We need to better understand the ecology to really move the needle and reduce transmission. And so the path to an R01, as I see it, is, again, I can't predict the future, but if it pans out, the kind of the hypotheses that I have are right, thinking about designing an intervention study to understand if intervening on networks improves malaria control. So rather than intervening here and having people come from here and reintroduce parasites, what if we intervene on the entire network? Can we do a better job at reducing transmission? And this K01 on top of that is allowing me to begin building necessary partnerships to hopefully translate a lot of my work into different practices. Again, if it turns out that the hypotheses are correct and the studies that we develop lead to improved malaria control. And so this fundamentally involves working with implementation scientists, which I've told a number of you already, I am not one. But I have found myself working in these settings over the last 5 or 6 years where we're developing a lot of tools and improving surveillance systems, and we're starting to ask questions about like, okay, well, is this useful, right? How do we translate this and get people to adopt, change malaria control practice or adopt a piece of technology, etc. And so one of the best pieces of advice that I had came from my primary mentor, Bill Pan, and he said, you need to put Valerie Custle down on your mentorship committee. And so she's a Peruvian-American behavioral scientist who has 2 big grants right now on implementation science in the Peruvian Amazon, one on cervical cancer screening and one on dengue. And she's really helping me think through a lot of the questions about, like, how do we start asking about, what are the barriers to implementing current interventions? So funding, issues of safety. So some of you may know much of the Amazon is cocaine production and trafficking territory, and people don't necessarily want to travel there to deliver malaria control. And then kind of the future questions are, all right, well, what are the barriers to implementing intervention activities, say, based on networks, if this grant pans out, etc. And so those are kind of the directions that I'm heading with this grant, answering these questions and hopefully translating those questions into improved malaria control in the Peruvian Amazon. And so just to wrap up, because I know we're probably short on time, I'm a geographer and a biostatistician. I work on human-environment interactions with the aim of improving malaria control. I've done a lot of work in the Congo. I still have some ongoing work there, some papers that I'm working on. But most of my work these days is focused on the Amazon basin and trying to understand the issues that are presented today. Gave you these 2 examples of my Congo and Amazon work. And then, as I move forward in this, I'll be writing an R21 or an R34 to kind of plan these intervention studies and then fund them via an R01 moving forward. So that's kind of not just the next 3 or 4 years, but hopefully the next 4 to 10 years as well. And so I just want to wrap up by thanking kind of a lot of the people that I work with. So I work in large multidisciplinary teams across multiple institutions with lots of different expertise. And so some of these people are on my mentorship team. So Bill Pan is kind of my primary mentor. He's an expert on the Amazon writ large and human environment interactions in the Amazon. Jim Moody is probably the most famous network analysis person, one of the most famous network analysis people on the planet, certainly the most famous one at Duke. John Giuliano is a malaria genomics guru and an all-around nice guy, and then Valerie is an expert on network analysis, the Amazon, Peruvian Amazon in particular, and implementation science. And so they've really been helpful in reading drafts of things and offering some good guidance. So with that, I'll thank you. I'm happy to take any questions.
Okay, we've got about 7 or 8 or 9 minutes. Any questions?
Yes.
Yeah, go ahead.
This was a really cool presentation, thank you. I was curious, like for your study DRC, did you take into account whether like children under 5 had like seasonal malaria chemoprophylaxis?
No, we didn't. So that's a good question. Probably should. I believe the data, actually, I'm not sure the survey data asks about that. It's been a while. I'd have to go back and look, but that's a really good one. Yeah. It could be like some of those spots, it's like just coincidentally there have been like some sort of seasonal chemoprophylaxis intervention that led to declines in transmission. That's really good.
So my question is about your key informant interviews. So it sounded like you had just a small number at each site, and I was just curious as to who the stakeholders are, thinking about the different genders and potential very different sort of pathways and movements of people of different socioeconomic status and gender.
Yeah, so we've done a pretty good job of making sure that our field teams are gender balanced and making sure that our key informants are also gender balanced. And when we recruited key informants, we basically went to the village or the community and we asked various people like who really knows the community well. So kind of people who've been around a while. And so when I was in the field, like one of the people that we were interviewing was a female business owner, restaurant owners, and so she just had tons of people coming in every single day to a restaurant, talking to them, and so she knew how people move. Similarly, in the same community, we were talking basically to kind of like the local, the person who grants marriage licenses and stuff like that, so kind of a government official who kind of knows the lay of the land as well. So we really sought out kind of a diverse group of people who kind of knew how the community was connected to others. I didn't go to all 300 communities, so I'm sure there's some variability in how well that was done. But we found that there's a kind of a high degree of agreement and kind of a good amount of variation in key informant demographics, and so we feel pretty good about that. In 6 months or so, I might have some better descriptive statistics. The data are just kind of getting cleaned and put into the system. But that's a good question. Early on in the study, when the field team was there, I noticed that there was, one of the Peru sites, there was kind of a gender imbalance in survey respondents. And so, fortunately, when life is on WhatsApp, you can say, hey guys, can you improve things a little bit and make sure that we have sufficient female representation?
And you use sequential referral anyway, right? When you were doing
Yeah.
Jim, this is Catherine. There's lots of excitement online as well. I just wanted to make sure that others online had a chance to ask questions.
You guys online, so thank you. Yeah, go ahead.
That's my fault.
Mark, I wanted to thank you for the presentation. It's fascinating. As a demographer myself, I love the interdisciplinarity that you bring to your work. I wanted to take a step back and to think a little bit about sort of big global population trends and what implications that may or may not have for malaria incidence and transmission. So we know that a majority of the people already live in urban areas, that urbanization is rapidly increasing, and cautionary projections appreciated, a majority of the world's population are going to be living in urban areas. Livelihood strategies are changing out of agriculture into other livelihood strategies with global increases in education. Can you talk a little bit about what the implications are for malaria with these big global patterns?
Yeah, so that's a really good question, and I'm happy to address it. So, big demographic trends. Africa's population is expected to double in the next 25, 30, 40 years. I don't remember the exact timeline, from about 1.25 to 2.5 billion people. And a place like the Democratic Republic of Congo that's really food insecure, and there's 100 million people, if that population doubles, we want to make sure that we're addressing that problem. Related to kind of urbanization, I think in one sense, we should see a decline in malaria as a function of urbanization because principally most malaria vectors really don't like big cities. They really like clean water. There is one exception though. Anopheles stephensi is an urban malaria vector that is predominant across much of South Asia, and it's now been found in sub-Saharan Africa, and East Africa in particular, in Ethiopia and Addis Ababa. So we're not really necessarily out of the woods through urbanization. There's this kind of mega trend of urbanization, you're right to point out, but there are also some implications for malaria if urban malaria vectors become more established where there's a high burden of malaria already. So those would be kind of my two answers off the top of my head.
Can I follow up quickly on that? Because my question was kind of very similar to Catherine's. You know, I was just kind of wondering when you were describing this, and thank you, by the way. I think Felipe and I no one has ever said soccer as many times in a presentation, so that was just like a beautiful thing. But I'm sort of wondering, because you see the stall that we've been having in malaria over those years, and I wonder if there is some kind of natural threshold. I mean, in a country like DRC that's got so much agriculture, right? Whether without really fundamental changes to the built environment, not just human migration to cities that already exist, but whether without some of those really fundamental changes in the built environment, whether that's just going to be a set level, or I guess the question was A, what you think about that, and B, how some of the work that you're doing may help us understand whether we're getting at something that's maybe a little bit of a sticky threshold there.
Yeah, I think that's right. I mean, I remember having a conversation with one of my mentors a long time ago, and I was kind of being jaded, and I was like, why do we keep doing all this research? And he's like, absent massive economic development where we fundamentally transform housing structures and stuff like that, malaria is still going to be with us. And I think that's to your point, is the environment as it exists now is really conducive to malaria transmission in the tropics. Where malaria has been eliminated tends to be in kind of more arid ecosystems and stuff like that, and so it's just a fundamentally different animal to eliminate malaria from the tropics. The Amazon and the Peruvian Amazon got really close. They were in this transmission. Ecuador is in fact on kind of the 2020 or the 2025 elimination list. I can't keep track of it because transmission is sufficiently low, except most cases, particularly of Plasmodium falciparum, are being imported from Peru. And what I would say is that fundamentally the interventions that we have available to us won't by themselves eliminate malaria. So there will have to be additional work, either changing the interventions themselves or supplementing them with some mass drug administration, or understanding the local environment better so that we understand when mosquitoes are biting and we're saying, well, maybe don't expose yourself to malaria-biting mosquitoes in some way at this time, etc. So my work is trying to inform on the latter. But I think you're right, that if the environmental conditions exist for malaria, absent a lot of human intervention work, it's going to be there.
Yeah. I think there are some comments online.
Jessica.
Yeah, hi there. Thanks so much. I'm sorry I'm not there in person. I got a sick kid that set my day a little bit, but I'm an infectious disease researcher, and thank you for giving me more for my soapbox that you can't rely on or that you have to integrate local context to understand better environmental and climate related effects on infectious disease. But my question is that given this kind of recent news in the DRC of this kind of mysterious disease in the south that's looking like it's malaria in the context of significant undernutrition and malnutrition. To me, it seems like we also might be missing, there might be other factors that your work could shed light on. How could you see this type of work that you do in the environment kind of help with outbreak investigations? Like maybe there was some kind of weather event that's now impacting nutrition, malaria risk, etc.
Yeah, so that's a really good question, and it kind of is related to some of my work in Madagascar. So I have a new project, and kind of a new collaboration since I returned to Duke, where I was working with a colleague of mine who's an evolutionary anthropologist, and they had a survey in the field asking about rural farmers' perceptions of climate change and asking about their adaptation strategies. So what are basically farmers are saying, yeah, the dry season is longer, my crops aren't as good, and I have to adjust my behavior in some way to kind of continue making a go at it. And one of the main adaptation strategies that they cited is they sell livestock. And so kind of in my mind, as someone who studies mobility and has been to Madagascar, selling livestock fundamentally requires, and it's mostly chickens and ducks and stuff, putting them into a tight cage on top of a bush taxi, a taxi Bruce, which translates to bush taxi, and taking them to a market. So you have basically kind of relatively free-roaming poultry, fowl, who are now crammed into a bus and then crammed into a crowded market to sell. And so that kind of gave rise to a grant idea where it's like, okay, well, this climate adaptation strategy as a function of the changing environment is going to change people's mobility patterns as they travel to sell livestock, and that can give rise to kind of emerging infectious diseases. So we actually just got a grant internally from Duke to look at this and basically take swabs of chickens and ducks and other livestock and humans to kind of understand if this is leading to kind of pathogen diffusion of flu viruses, for instance. And then in the same area, we just kind of discovered, and I know nothing about this, I've just been on the Zoom meetings, but Orf virus, which is a virus apparently in animals, has been detected in some of our human samples. And I'm only aware that there's lab work trying to confirm it and stuff like that. But this is kind of work that we're starting to understand. It's like, how are these contact patterns perhaps changing as an adaptive strategy to climate change, et cetera. And I think that's kind of related to what kind of you're getting at. And maybe that's kind of what's playing out in the Congo with kind of quote unquote disease X, but I'm not on the ground there right now, so I don't have a lot of insight.
Can I ask you a really specific question, Mark?
Yeah.
The human landing catch approach, which I've actually done as well, I've done a lot of mosquito stuff, is that just thought to be a better proxy for biting rather than, say, traps?
So that's how I think of it as a non-entomologist. And when the entomologists that I work with, I've been in the field and we've tried Shannon traps and we've tried the Procopex and we've tried human landing catch. And so far we've collected more mosquitoes with human landing catch. But we can also vacuum them up from the inside of surfaces and they're resting. And so it's like, well, we might assume that if they're resting inside the house that they bit inside the house. So there are many ways to do it. And each has their biases and stuff like that. And so it's just kind of one of the issues that I kind of think about, but kind of ask my entomology colleagues about, so that I don't make a fool of myself trying to be an entomologist.
But it's hugely labor intensive compared to trapping.
Yeah, it's really labor. Sitting out all night and just every 30 seconds turning on a flashlight to look at your feet.
And scary.
Yeah. And then it's not the malaria I'm worried about getting, it's something else.
Jim, I have a quick question.
Usha,
I may? I know we're past the hour.
using the
So,
privilege of the chair.
this is a more question. It's nice to hear the word geography. There's generations who don't know what that means, or younger than me. But my question is, you talked about being a biostatistician, and when we talk about implementation science, there is this, mixed methods. How do we triangulate qualitative methods with, the high-flying statistical analysis? And maybe I missed it because I came a few minutes late, but I was just interested, as you talk about network analysis, neural network analysis has been around for a very long time, used in finance, used in other areas. We have some faculty here who also have statistics who use machine learning. I just wanted to get your thoughts on that approach. And is it just like you said, something different called, the same thing called by a different name?
Yeah, I think there's a lot of that. Like just between geography and biostatistics, there are two different ways of understanding spatial processes.
Don't forget the economists. But
Yeah, I've worked with a few economists who I really, really like, but the language is just so different. And so kind of, for me personally, like there are, a few things that I'm really good at. So I'm a geographer, I do a lot of field work, I can design survey instruments, and I'm a statistician, and I'm kind of branching out to network analysis and kind of population genomics. And then kind of for a lot of the other work that I do, kind of, for instance, in the climate sciences, I collaborate closely with climate scientists, I collaborate with implementation scientists. So, I believe that there's value in all of it, but I believe there's limited value in trying to do all of it yourself. And so I don't know if that gets to kind of the way you asked it, your question.
Well, I was thinking sort of the methodological innovation with machine
You're talking
learning.
more like social network analysis
Yeah.
rather than neural networks.
Okay,
And
yeah.
where do that twain meet? I mean,
Mhm.
Yeah, so with, I do a lot of machine learning kind of on some of my collaborative projects, and I was skeptical, just because it's like, well, machine learning emerged out of the computer science literature, and computer scientists really are terrible at characterizing uncertainty, and I'm a statistician, and that's what my life is about. I'm not sure about anything. And so, I was kind of curious as to whether or not, or skeptical of its utility, because it's like, okay, well, maybe it can make me, click add to cart, but like, what can it do beyond that? And I'm starting to come around and see that there's actually a lot of value in it. So I was just in Peru a couple months ago presenting some of our collaborative work to the Malaria Control Program, where we were essentially forecasting malaria transmission rates,
that's
6-9 months in advance, kind of trying to transform surveillance from reactive to proactive. And initially I was mortified because the machine learning algorithms, it's like, okay, we're going to select all of the different factors that are relevant for transmission in spatially explicit ways. And I personally don't think like that, because I'm just like, well, the machine is going to tell me what the risk factors for malaria are. I'm a little worried. And so I was presenting these results to the Ministry of Health, or the Malaria Control Program rather, and they're like, oh wow, that makes a lot of sense, because the river runoff in this section really is really bad, because then the rivers recede and it leads to these breeding pools, and then over here, deforestation is happening, and it's leading to a lot of increased transmission. And so I think, the success of that, or what I felt was the success of that meeting, was a combination of like, you can have garbage in, garbage out, but if you put some good data in, some good results might come out. And then, talking with people to contextualize them. So, I'm open to kind of different methodological tool sets to answer different questions, and I have varying different degrees of expertise in different things.
I mean, it's also building the kind of training logic, right,
Yeah.
for AI, where it's going to have to be long and elaborate, but I mean, what you're doing is adding those elements that often get ignored.
Yeah.
We've done some work with Lance Waller
you should meet Lance.
in Haiti, where my team has done a lot of like intensive interviewing around issues related to avoidance of mass drug administration, and then we've incorporated them in. Thanks for coming, Grace. We've incorporated that with some of Lance's mapping, so we're able to actually show areas, for example, we have low coverage in MDA, and then we drop pins exactly into the geography, because we have health surveys, like we have household surveys to anchor them in GPS, and we drop in kind of explanatory interview data right into the geography. So you look at it and you say, I wonder what the hell's going on here, and then we'll say, well, these 5 or 6 things could be part of the explanation of what's going on there. So it's kind of, we can talk about it later, but but some of
That's
that,
what I was getting at. I mean, it's in that mixed method.
it's the integration of it,
toolkit.
Yeah.
But I think there's a lot of.
Yeah, it's still tricky stuff to land, grant-wise, but there's some really promising stuff.
Yeah.
We're over time. Can I ask one other thing, since we're just going on anyway?
Of course.
I'm gonna run.
What about we've got a lot of phones that exist already are already satellite ready, right? And I wonder, have you thought a lot about next waves, like maybe 5-year waves, about when we're really going to be able to get better satellite thing, and maybe where Elon Musk won't hold the Starlink keys,
Yeah, so we actually had a grant in Peru that got funded. I wasn't the PI, and it was an internal grant to basically do that. And then there was a coup in Peru, so we had to switch countries and switch the grant idea to something else. But yeah, we've definitely thought about doing that. Yeah.
Okay, Mark, thank you so much, and thanks for hanging in for so long for all the questions. I'm not sure if anyone's still online, but thank you so much, anyone that is, and thank you guys for all coming in person. and really appreciate the presentation.
Yeah, Jim, quick comment before you close the Zoom.
Catherine, yeah, thank you.
Yeah, no, I just wanted to say thank you and also to recommend that Mark meet Weiwa An in the Department of Sociology, who's the editor of Sociological Methods and Research and does a lot of statistical work on social network analysis and causal inference. So that would be a great meet, I think, during the visit.
Could you
Or
tell
after.
me the person's name, Catherine?
Yeah, I'll send it to you. Yeah. Thanks, everyone.
All right, well, thank you, everybody. Thank you for coming.
Thank you, Mark.
Thank you.